Now we'll hear from Professor Sam Breit from the University of New South Wales, whose work on the MYC1 protein could potentially help us to diagnose cancers earlier.
Thank you for the opportunity to talk to you, and thank you for the acknowledgement of our work in this area. I'm not going to use slides, just to be a little different. We've been studying a protein called MYC1 for many years. It's a TGF-beta family protein, an unusual protein from this family, and we've been studying it since we first cloned it and characterised it in the 1990s. Now, it's been particularly linked to cancer. So when we wanted to study its role in cancer, we engineered cells to overexpress it, and we grafted those cells into mice, immunodeficient mice. And I guess, as often happens in this situation, we made some discoveries that were really quite fortuitous. What we saw was that the mice grafted with this tumour, but not controlled tumours, lost a dramatic amount of weight. They were healthy and happy, but they lost a great deal of weight. And we could tell that the amount of weight they lost was proportional to the serum levels of this protein MYC1, because we'd previously developed an immunoassay for it. Not only that, we could reverse it with antibodies specific to MYC1, and we could reproduce it by giving the mice the purified recombinant protein, which we'd also expressed. So to a clinician scientist like me, what these mice developed is really very much like a condition called cancer anorexia cachexia, or anorexia cachexia that's seen in a number of conditions like cardiac failure and in renal failure. People lose a very dramatic amount of weight, and usually this has a great deal of effect on their quality of life. And in the context of cancer especially, makes it impossible to treat them and speeds their death. I'm sure that most of you will know someone, often a close friend or relative, who suffered from this complication, for which there's really currently no effective treatment at all. Now, we'd been the first to describe that MYC1 was overexpressed in cancers. And it's overexpressed very commonly. And we'd been using an immunoassay we'd developed to try and define its usefulness both in the diagnosis and management of cancer. Now, from this, we knew that serum levels of MYC1 rose dramatically during the course of cancer. And in advanced cancer, they can rise 200-fold or more. And that's well within the range of the effects we were seeing in the mice. So it seemed like it was very likely to be incriminated. And really, to try and define this a little better, what we did is we found a cohort of patients who had advanced prostate cancer and really had been studied for other cytokines. We got some serum from these people, and what we were able to tell was that the amount of weight these patients lost over a 6-month period was proportional to the level of MYC1. The higher the level, the more weight they lost. And that really fairly solidly linked this particular protein to cancer cachexia in humans. Now, we still didn't know the mechanisms at that point in time, but what we did notice was that these cachectic mice ate very little. We could tell because there was much more food left in their, in the cage when it came around to changing. And it seemed like decreased appetite was a very likely mechanism, and ultimately that proved to be the case. We could show they ate less. That mice with this tumour ate less. When we gave them antibodies, they ate more. And we could show the same thing with recombinant protein. We then went on to do a lot of other studies to define the effects, and were able to show that this protein accessed parts of the brain that had a semi-permeable blood-brain barrier, and were able to directly affect feeding centres in the brain. So what this suggests, what this also suggested is that MYC1 was identifying a previously unknown pathway of appetite regulation that might be harnessed for a number of different purposes. So it might be harnessed for treating obesity, severe obesity, which because of its really very good behaviour in experimental animals. And that certainly seems as if it might be possible because we've been able to license this to Novo Nordisk, who's a Danish-based multinational that specialises in protein therapeutics, and they're especially interested in the metabolism area. And we expect there'll be first in human trials of this protein at the end of 2012. I guess the next thing it's suggested, of course, is that monoclonal antibodies to this protein may be able to be used to treat patients with this type of cachexia. And we could identify that subset of patients, because I don't think all patients with this type of complication, it's not going to all be due to this protein. We can identify that because we have an accompanying diagnostic that can identify those people who have levels high enough for that to be a realistic option. I guess that also looks as if it may well come to fruition, because the NHMRC have funded a development grant, the resources from which are going to Dr. Dan Christ at the Garvan, who's humanising the monoclonal antibody for St Vincent's Hospital, and we're planning to use this in clinical trials. I guess the last implication of this work, or the last thing I think it did, is that it altered the paradigm of cancer cachexia. When I first started talking about this issue, there was general disbelief, because dogma within the area suggested that whatever was happening was having a direct effect on muscle mass. It really had nothing to do with appetite. And there was a general view, incorrect, that if you stop eating and you lose weight, that you lose fat before you lose muscle. Well, the metabolism and endocrinology people have known for decades that that's not the case. You really lose both. And that really changed, I think is starting to change the thinking about how this process might be managed, and the potential role of other proteins that might also be overexpressed by tumours and that might act on appetite and other relevant centres. Our current studies are really focused on trying to understand the biology of MYC1 in cancer, and really to further understand the neural pathways through which it works. Now, a large project like this couldn't have succeeded without a lot of support from the NHMRC, from my institution, St Vincent's Hospital, from the Cancer Council, and the University of New South Wales. This project was also successful because of help from lots of collaborators, and there are really too many to cite. But I'd like to specifically acknowledge some of the people in my lab who were involved, and that's Heiko Jonen, Tamara Kufner, Vicky Tsai, and David Brown, and Herbert Herzog from the Garvan, who was really instrumental in defining the neural mechanisms underlying the effects of MYC1, and within his group, Amanda Sainsbury and Shu Lin. Thank you.